Thanks for joining us, and I'd like to introduce Julie Douville, who's our Executive Director of Drug Discovery and Development, to really pull back the curtain on how we make these optimized ASOs that we talk about.
Thank you, Amy, for the introduction. Hi. Today I'm going to talk to you about how at n-Lorem we use our ASO experience and scientific excellence to drive our discovery and development platform. I know that not all of you have a scientific background, so hopefully I can do this so that you can follow what I'm saying. So let's start at the very beginning. Genes are made of DNA, and DNA is made of 4 building blocks that we call nucleotides or bases. And we refer to them by their symbols, A, T, C, G. And these nucleotides are organized in a very specific sequence, a very specific order, so that information can be encoded in our genes. And from there, this information is transcribed into messenger RNA, which really bears the instructions to make a protein. And proteins are complex molecules that are really doing all the hard work in the cells, and they're important to the structure, the function, the regulation of the body. So let's talk about mutations now. What's a mutation? A mutation essentially is a change in the normal sequence of nucleotides. I heard someone call it misspelling this morning, and I really like this analogy. And so it can be a deletion or an addition of a single or longer stretch of bases, but it can also be the substitution or replacement of one base for another in the sequence. And more specifically, we refer to these as SNP, SNP, single nucleotide polymorphism, and I'll talk about that a little bit later. It's kind of important when we design ASOs. And most of the time, mutations are either corrected or they're benign or silent, which means that they don't have any consequences. But other time, occasionally, mutations can change the critical part of the instruction to make a protein. And so as you can imagine, different types of mutation will have different consequences. For example, a mutation can cause a protein to become inactive or be degraded by the cell. So the protein is no longer able to do its normal job, and we refer to these as loss of function. Other times, a mutation can cause a protein to acquire a new function, so a job that it wasn't doing before. And these mutations are referred to as gain of function, and often they're toxic. And so when we get an application for treatment at n-Lorem, it's really important that we do a thorough evaluation of the nature of the mutation, its consequences, and so that we're able to come up with an adequate ASO strategy for each patient. As you can see on this chart, a lot of our patients have a gain of function mutation, and so the rest of my talk will focus on this type of mutation. Gain of function mutation can be addressed by RNase H1 ASOs. So those are ASOs that will allow us to downregulate a gene. Many of our patients will require an allele selective strategy. And so Stan talked about that this morning. And we need to use this approach when the function of a gene is essential to the cell. And so we need to be able to knock down the bad copy while maintaining the good copy. And as you can imagine, it's a lot more difficult to come up with an ASO that will have this level of selectivity for basically a difference in sequence that is only one base. And so I'll cover that in more details later in my talk. As we discussed this morning, our platform is based both on ASO technology and FDA guideline. So Stan talked about that, how this technology has been validated. We know a lot about it, and it's also versatile. So we can change the sequence to target a different gene, and we can predict what's the dose that's going to be efficacious. And we can also predict adverse event quite well with this technology because we know so much about it. It's also cost-effective, which makes it scalable. And importantly, the FDA, with those 4 guidelines that were issued, really provided a roadmap of what is needed to advance these molecules to the clinic in a very streamlined and efficient manner. This slide here is just to show you a number of articles that our group, STAN, has published recently on how RNA-targeted drug discovery can be applied in the field to address the needs of the patient. And so I encourage you to go and do some reading if you want to learn more about the technology. And now we're going to switch gear and cover our process to discover and develop an optimal RNase H1 for a patient with a CNS disease. And so each of these steps are critical in coming up with the best possible ASO. And as Stan mentioned, we can't skip a step. And the minimum criteria that we use at each of these steps, as well as the number of ASO at each step, is also really important and based on decades of experience, as well as thousands and thousands of ASOs that have been screened over the year. And so now I'm going to cover every step in more details. The first step is, of course, to design the ASOs. And I'm going to focus on allele-specific ASOs here. So for each gene, of course, we have 2 copies, one that come from our mother and one that come from our father. And the pathogenic mutation, so the mutation that is causing the disease, can be present on either allele. So for the sake of this example, we're going to put it here, the red X on the top allele. And so the messenger RNA that will be made from that allele will be referred to as the pathogenic mRNA because it bears the pathogenic mutation. And the other messenger RNA will be referred to the wild-type mRNA. So in addition to the pathogenic mutation, we can have benign changes in bases. So those SNPs that I mentioned at the beginning, they can be present on either allele. And depending on the gene, there can be many SNPs or there can be fewer. It's really depending on the gene and on the individual. And so they're identified here by the black X's. And these are really important because each of these sites provide us with an opportunity to design allele-specific ASOs. Because at these specific sites, the sequence is different on both allele. And so we can use this to design ASOs that will recognize or bind just to one copy of the mRNA. And in addition, we can also use the sites that are opposite. So the sites on the pathogenic mRNA that have the normal sequence, but that are opposite to a SNP, because again, at this site, the sequence is different. And so we can design ASOs that will be specific for the pathogenic mRNA. And so ideally at that step, we want to have designed between 400 and 600 ASOs. And we like this number because when you start with such a large pool of ASO candidates, then it gives you the best chances to identify the best ASO. And so I'm going to explain to you now how these ASOs are working. So you have the pathogenic mRNA there with the pathogenic mutation, as well as the wild-type mRNA. So our ASO will bind here where the sequence is unique to the pathogenic mRNA. And this will create a duplex of ASO mRNA. And this duplex is recognized by an enzyme that we call RNase H1. And this enzyme will cleave the duplex and trigger the degradation of this duplex. And so there won't be any mutant protein made from that mRNA. On the other hand, on the wild-type mRNA, the ASO is not binding. There is no recognition by the RNase H1. Therefore, we can maintain production of a healthy protein. So this is how we can make allele-specific ASOs. And so once we have designed these ASOs, the next step is to test them for potency. So potency is how effective these ASOs are at knocking down the expression of the pathogenic mRNA or reducing, basically, expression of the pathogenic mRNA. And so In this example, we're looking for an 80% reduction of expression. So I draw that purple line where the threshold is. And on this curve, each point is a different ASO candidate. And so in this example, we would take these ASOs in the orange box and move them forward to the next step. The next step is very similar. We're looking at potency once again, as well as selectivity for the pathogenic mRNA versus the wild-type mRNA. And so you have your pathogenic mRNA here in red, and the wild-type is in green. And what we're looking for is a dose-dependent reduction of expression, because we want to understand that when we increase the dose of the ASO, we have a dose-responsive effect on the expression. So that allows us to predict, with whatever dose of ASO, the kind of effect we're going to have downstream. And so in this example here, really what we're looking for is, as I mentioned, a reduction of the pathogenic and a maintenance of the signal for the wild-type expression. And so, for example, this ASO number 7 here, we would move it forward to the next step. We would probably also move ASO number 3 forward, and maybe even ASO number 6. Not 6, 5, because the effect on the wild-type expression is not significant compared to the pathogenic mRNA. And so at this stage, we're now looking at an assay that we call the BJAB assay. So essentially, that's an assay that is designed to measure activation of the innate immune system. And so some ASOs are known to trigger an inflammatory response, and that's not desirable. And so this part of the screening is really designed to identify those ASOs that would trigger this type of response. And basically what we're doing is treating peripheral cells. So the cell line is called BJAB. That's where the assay takes its name from. And we're treating these cells with all of our ASO candidates that we have moved forward. And we're basically comparing them to 4 control ASOs that we know are either highly inflammatory or moderate, or basically not inflammatory. And so those are the green, blue, yellow, and red lines on the graph. And we're really benchmarking our candidate ASOs against these 4 controls. And in this example, ASOs that are in the orange box would move forward to the next step because they're considered safe. The next step is looking at potential off-target effects. So this is really looking at whether there's any non-specific binding of our ASOs to messenger RNA from other genes. And this non-specific binding of ASO could, in theory, reduce expression of genes that are critical for the cell. And although it's extremely rare to have toxicities that are related to off-target effect, we still want to do our due diligence and really look at this in more detail. So it's a 2-step process where the first part is a computer-based analysis where really we're going to highlight any genes that would have a partial match with the ASO sequence. And then once we have identified those genes, we will run an in vitro assay where we're going to use cells, treat them with our ASO candidates, and then do PCR to evaluate level of expression of those potential off-target genes. On occasion, we might have a slight effect, minor effect on these genes. And at this point, it's really important to have a really good understanding of our patient condition to really be able to do a risk-benefit analysis here and understand if there's a true liability of this effect. And so once we've completed that step, ideally, we are probably around, down to around 20 ASO candidates. And so we'll take these 20 ASO candidates and we'll do a small batch synthesis of research-grade material. And we're going to have, even though it's a research-grade batch, we still want to have strict criteria for purity and other aspects as well, because the next step will involve animal testing, and it's important that animals are exposed to a batch of material that is comparable to what the patients will receive. And so the first animal study that we're running is a tolerability study. So this study is not designed to look at efficacy, but really designed to identify poorly tolerated ASOs. And we use a route of administration that will be similar to the route used in the clinic. And the animals will receive a single dose at quite a high concentration of those top 20 ASOs from the in vitro screen. And we're going to evaluate the animals over a period of 8 weeks. And we look for any abnormal clinical signs, any effect on neurobehavioral parameters. We look at their body weight. And then once we reach that 8-week period, animals are taken down. And we collect tissues of interest. So for example, a CNS target, we would collect spinal cord and brain. And we look for specific markers of inflammation and toxicity. And then from this step, along with all the data that we've accumulated from the very beginning when we started the first, the single dose potency assay, and then the dose response curve, and the BJAB and the off target, we take all of this data, look at it in the context of the patient, and identify which ASO has the best potency, the highest selectivity, and is the best, the best tolerated ASOs. And that's the one that we're going to move forward to become our clinical candidate, basically. And so at this point, we're going to have 2 steps that will run in parallel, just because that's how we can optimize the timeline. And we'll move into GLP toxicology study. So this study is really important. It's the one that allows the FDA to approve the treatment, and it's designed basically to look at toxicity after repeated dosing over a chronic period of time. So this is designed really to represent a situation of a patient receiving a drug over a long period of time. And the specific design of this study will change based on the organ that we're targeting and the ASO strategy that we're using. But what's important to understand is that this study is strictly regulated. All parameters are controlled. It can be, the conditions in the animal room, the temperature, the humidity. It's the temperature in the freezer where we keep the samples, where we keep the drug. It's how technicians are trained to perform that study. Really, all parameters are controlled, and this ensures that the data that is generated is robust and reproducible. And in this CNS example, animals will be injected once a month by intrathecal injection, and we will look at them over a period of 3 months. And some parameters that are evaluated are general, such as body weight, food consumption, but other parameters will be specific to the organ that we're more interested in. And so it would be neurobehavioral assessment here that we do on a regular basis over the course of the study. And then at the end, animals are taken down. We collect tissues to look for any abnormal pathology finding. And we also look at drug level in those tissues to understand the distribution of the drug. In parallel, we'll trigger GMP manufacturing of our drug. And so similar to GLP tox, GMP manufacturing is a regulated activity. It operates by strict rules to ensure quality, reproducibility at every step. And we're going to have, once again, very strict criteria for purity, salt content, sterility, endotoxin level, to make sure that the drug is suitable for human administration. As we mentioned before, single batch of an ASO manufacturing provide enough material to treat a patient for 5 years, 10 years, or even more. So it's very cost effective. And then depending on each institution, so where our patients are going to be treated, we might need an additional manufacturing step, which is basically taking this powder ASO, formulating it in a liquid form, sterile, that will be aliquoted in vials for administrations. This is called sterile fill. So the FDA guidelines have outlined a fairly lean, non-clinical pathway, fairly straightforward, which makes the timeline a lot more interesting for Nano-rare patient. And that's why the rigor and quality are of the utmost importance at every step. As you can see, non-clinical data represents 60 or even more, maybe 75% of the content of the IND, and is really a pivotal part of getting that drug to the patient. The rest of the IND is really focused on the patient. At n-Lorem, we're very patient-focused, not population-focused. So the rest of the IND is around our patient and a clinical protocol around it, and Lori's going to cover that in more detail right after me. And so we have developed a mutation-driven discovery and development platform that is scalable while at the same time being tailored to the needs of each patient. As you can imagine, this requires very deep ASO expertise and experience, as well as agility and flexibility in our decision-making process. And I'm missing a slide. Sorry. Okay, and so really what is the most critical is to integrate decision from top to bottom. So each step of this process is extremely difficult, but integration throughout the process is even harder. And we really perform this risk-benefit assessment in real time as our ASO program progresses, but as well as the patient phenotype also potentially progress. And so essentially, my slides are in the wrong order. Essentially, the most important question that we ask ourselves at every step of this process is whether this ASO is adequate for this specific patient. This is really the most important question that we ask ourselves all the time. So thank you for your attention. Thank you to all of our supporters and partners in the industry. We couldn't do that without you.